So hello, very good morning. I hope my voice is audible and the video is visible. So please let me know if it is fine or do I need to adjust anything. Okay, fine. We will wait till it's 11 a.m. and then we will start today's session. Okay. So very good morning. We will wait till it's your 11 a.m. and then we will start today's session. Meanwhile, let me know if my voice is audible and if you can see the presentation clearly, or if there is any problem, let me know. So, let us wait till it is 11, then I will start the session. Okay, I think I will start with the introductions. Okay, it's almost 11 a.m. So, just let's start with the introductions till other students join. Okay, first just make it open. Yeah. So, very good morning to all of you. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our summary session. So, this is in this 1 hour summary session, what I will do is I will summarize few things about your course. So, basically what every day I do is, like, whatever the week is live that time, I just take around 20 questions from that week, particular week topics, and then we discuss that 20 question. We solve some MCQs and then I discuss. So that is how we just proceed with our problem solving sessions. Today's class will be a little bit different. So, what I will do is today I myself will summarize few things of medicinal chemistry or the topics of biochemistry. Okay, these topics were involved in your course. Okay, and in initial weeks. Okay, and these topics, some few topics you know very in detail, and few topics that wasn't in very detail in your course, but these topics are important for medicinal chemist. So, I thought it will be better if I can give you a brief about the topics which were not discussed very widely in your course, but that are important for medicinal chemistry. So, today in this 1 hour session, first I will summarize the topics. Okay, and after that we will have a few questions, few MCQs. I will give you around, I think 5 to 6 MCQs, I think 5 MCQs, which like every day I will give you 4 options, you have to solve and you have to answer, but that we will do at last in our toward the last in our session. But first what I will do is I will summarize few things. Okay. During the session, if you have any doubt, any query, as I have already told you that you can just post any doubt, any query, everything in the chat box. Okay, I am checking the chat box always. So, if you have any doubt and any query, just let me know in the chat box. And at the end of the session, like always, I will share my email ID. So, if you have any problem, any doubt, you can share with email too. Since we are almost at the end of our problem solving sessions, so today's class and the last class we will be having on Tuesday, on the day after tomorrow, which is on the 16th April, Tuesday from 6 p.m. to 8 p.m. That will be our last class. That day I will also summarize few topics of medicinal chemistry, and we will have few question answer sessions. So, that will be our last class. So, if you have any problem or any problem in any topic on the course, please let me know by today or tomorrow, so that I can clarify that in our last class. Okay. And I have like always, I have muted the microphones and also the camera, so that I can record the session. Okay. Since I am recording the session, if you have any problem with internet, you can just leave at any time and rejoin at any time. Okay. I am recording the session and I will share the recording with you soon. And I think I have shown you where to find the problem solving session recording. If you have any doubt regarding that also, you can let me know at the end of the session. I will again repeat that. Okay. And this presentation and recording I will share to you. And one more thing, if there is any network problem from my side, okay, if it is just the disconnection happens at any point, do not leave the session. Okay. I will try to rejoin as soon as possible, so do not leave. So, I think I can start the session. The chat box is open. If you have any problem or any doubt regarding any topic, please let me know. Okay. First, I will summarize the topics, and then after some time I will give you few questions that will be MCQs, how we do at every day in our day to day class. So, you have to try to answer the questions, and I will show you the answer, and we will discuss those things. Okay. So, let us start our summary session of the medicinal chemistry course. Okay. So, the first topic which I will go through is the amino acids. So, amino acids are called the basics of life, right, because all the proteins are made of the amino acid. The amino acids are the simplest unit of our proteins. The amino acids, they form the peptide bond which goes to the primary structure, secondary, tertiary, and a quaternary structure which give rise to the protein. The amino acids is a very important component of our day to day life, and also in the all the points of medicinal chemistry and chemical biology also. So, what are the amino acids? With some there are around 20 to 22 amino acids, in which the 20 are naturally found in our body, and there are few amino acids which are not found in our body, which we have to take with the food, and few are the unessential or non-essential amino acids also. So, essential amino acids are those amino acids which is not formed in our body. Okay. We have to take these amino acids via food. So, what are these essential amino acids? These are the phenylalanine, valine, threonine, tryptophan, methionine, leucine, isoleucine, lysine, and histidine. Okay. So, this phenylalanine, it is crucial for the neurotransmitter production, impacting brain function and mood regulation. Valine is crucial for muscle repair, growth, energy, blood sugar, and immune system. Threonine is crucial for bodily functions, nervous system, health, collagen, elastin, and tissue maintenance. Tryptophan, it is important for serotonin production, mood regulation, niacin synthesis for energy and cellular health. Methionine is crucial for methylation as a precursor of antioxidant and detoxification. Then we have leucine, which is crucial for muscle growth, growth repair, the repair, regulating blood sugar, providing exercise energy. Isoleucine for muscle repair, endurance, regulating blood sugar during exercise. Lysine for growth, tissue repair, and healthy immune system. And histidine, it is for histamine production, which is the precursor key component of the physiology. So, these 9 are the essential amino acids, and I will tell you to remember these amino acids, and also along with their structures. Okay, the structures of this essential amino acid, because at some point or other you will need it. Okay. Then classification of amino acids can be done via several techniques. Okay, we can have a charged amino acids, then we have polar, non-polar amino acid, positively charged, negatively charged amino acid, and so on. So, the classification of amino acid according to the polarity of side chain, which is can be like this. So, what is the basic structure of amino acid? Like we all know, the basic structure is we have a amine group here, right, and then we have a carboxylic acid group. And with this alpha carbon we can have R1 or R dash. Okay. So, based on these two group R dash, based on the polarity of these two group, we can have different amino acids, the classification can happen. Okay. So, the major classification depending on the polarity will be the polar amino acid and non-polar amino acid. So, polar amino acids are those in which the R contains a polar hydrophilic group. So, it can form hydrogen bond with water. In those amino acids, R may contain a hydroxyl group OH as in serine, threonine, tyrosine. It can have a SH group as in cysteine, amide group as glutamine and asparagine. NH2 group nitrogen act as a base and in basic amino acid like lysine, arginine, histidine, and carboxylic acid group in acidic amino acid like aspartic acid and glutamic acid. Then we can have a non-polar amino acid in which the R is the alkyl hydrophobic chain which cannot enter in hydrogen bond formation. 9 amino acids are non-polar like glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline and methionine. So, these are all the non-polar amino acids which have R as a alkyl or hydrophobic group which cannot participate in hydrogen bonding. This polar non-polar amino acids classification is very crucial because sometimes it happen you will hear this term very frequently that there is a mutation of point mutation. So, what happens during the mutations, if you see this is a more important your example is like in sickle cell anemia. So, there so what happens if one polar amino acid is replaced by a non-polar amino acid. So, what will happen like the polar amino acid it can form hydrogen bond. So, it can interact with hydrogen bonding with the nearby amino acid residues, but if there is no hydrogen bond in case of it, if that particular polar amino acid which is stable in a particular protein network, if it is replaced by a non-polar one, so what will happen, it cannot form the hydrogen bonding now. So, what will happen, there will be the disruption of hydrogen energy and there will be abnormalities, and this is a very important example is like in sickle cell anemia. If you read the sickle cell anemia or if you remember, you can just identify that this is the polar non-polar amino acid interchange that is happens during the sickle cell anemia. Then we have the fibrous protein and globular protein. So, depending on the what is the size or what is the shape of the particular proteins, the protein can be divided into fibrous protein and globular protein. So, what happens in fibrous protein, it is a class of insoluble proteins which constitute the main structural elements of our body, and the globular protein, a protein soluble in water, tend to involved in metabolic functions. The structural protein has a long narrow shape or have a helical and sheet structure, but the globular protein has a round and spherical shape and have a folded ball like structure. The fibrous protein in the secondary structure and the globular protein in the tertiary structure. The fibrous protein consists of repetitive amino acid sequence, whereas the irregular amino acid sequence can be noticed in globular protein. The fibrous protein is insoluble in water, acid and bases. The globular protein is soluble in water, acid and bases. They have a strong intermolecular interaction in case of fibrous protein. In case of globular protein, they have weaker intermolecular interaction. In fibrous protein, less sensitive to changes of temperature and pH, but the globular protein are sensitive to the changes of temperature and pH. The fibrous protein involved in forming structure, globular protein involved in metabolic processes. The example of fibrous protein is collagen, elastin, actin, myosin, fibrin and keratin, and the globular protein is example is enzymes, hormone, hemoglobin and immunoglobulin. So, that are the examples of fibrous and globular proteins. So, how to calculate the isoelectric point is one of the important topics regarding your amino acid thing. So, this was not very in detail, but the questions can be asked from it also. Even if you not for only this course, if you go to any kind of competitive examinations for chemistry, you will find this kind of your isoelectric point calculation topic. So, that's why I thought to just give you a brief about it. So, what is the isoelectric point? So, we all know that the typical amino acids are deuterion at neutral pH. Deuterion means the positive and negative charge thus stays in a same molecule. An internal acid base reaction between the basic amino group and acidic carboxylic group result in a salt with 2 point charges, but net charge is 0. Typical amino acid have 2 acidity constant, one for carboxylic acid and one for ammonium salt. Okay. So, this is in the acidic condition, we have the both amine group and carboxylic acid group as a protonated from. Then the first decarboxylation happens from the carboxylic acid and we get the deuterion where the net charge is 0. And then we have this anion basic charge where the net charge is -1. Okay. So, this is the K1 and this is the K2. If there is the isoelectric point of an amino acid is the pH in which it bears net 0 charge. For amino acid with non-acidic or non-basic side chain, the isoelectric point or Pi can be the average of 2 pKa values. Okay. The Pi is the pK1 plus pK2, the pK of carboxylic acid or the pK of to ammonium ion. So, Pi will be the average of the two pK values. Okay, this will be Pi equal to, this will be average. Okay, this will be by 2. So, the Pi will be the average, means this by 2. Okay. And then if the amino acid, it has a particular acidic or basic side chain, in that case we have 3 different pKa values. Always you have to remember that the zwitterion will be that point when the net charge will be 0. So, typically in case of acidic amino acid, it is obtained by the averaging to most acidic pKa values, and in case of basic amino acid, you will obtain it by averaging the two least acidic pKa values. So, that is how you have to calculate the isoelectric points of the amino acids. Okay. Then we have the enzyme kinetics. So, enzyme kinetics is a very important topic of your medicinal chemistry. All the bodily activities are regulated by different works of the enzymes. We all know the kinetics of an enzyme, it helps us to identify that how it happens, okay, what is the particular concentration of enzyme which is required, and so on. So, what is the enzyme kinetics? So, basically the study of the rate at which an enzyme works is called the enzyme kinetics. Okay. So, we have substrate, we have enzyme, these are the smaller ones are the substrate and bigger one the enzyme, and we have the, this is the low substrate concentration and this is the high substrate concentration. So, reaction rates increase as the substrate concentration increase, but became saturated at very high concentration of substrate for a given enzyme concentration, and for relatively low substrate concentration, the reaction rate increases linearly with the substrate concentration. However, at relatively high substrate concentration, the reaction rate asymptotically approaches the theoretical maximum. The enzyme active sites are almost all occupied, and the reaction velocity at this point is represented by Vmax. The substrate concentration midway between these two limiting are denoted by the Km, which is the Michaelis-Menten constant. So, this is the equation of enzyme kinetics. First, we have the enzyme and substrate, and the equation goes to the enzyme substrate complex. The forward reaction constant, rate constant is K1, and the backward is K minus 1, and then we have this K2, which is which gives us the catalytic step rate constant, and which gives us the enzyme and the product, and this is the graph. In the y axis, we have the reaction, this is the reaction coordinate, okay, and this is the s axis, which is the substrate concentration. Initially, at the lower concentration of substrate, we have the first order kinetics, in between we have mixed order kinetics, and a high substrate concentration we have a zero order kinetics, where the enzyme kinetics or the velocity of the reaction does not depend on the substrate concentration. So, for a simple single step enzymatic reaction for conversion of substrate to product is as follows, where K1, K minus 1 and K2 are the rate constant for the individual steps. So, a key assumption is the derivation of the QC steady state approximation, namely that the concentration of the substrate bound enzyme and hence the unbound enzyme change much more slowly than that of the product, okay, and substrate. That of the then those much product and substrate. So, we can say that ddt of ES equal to 0, and which equal to K1 E into S, concentration of enzyme and concentration of substrate minus concentration of ES in bracket K minus 1 plus K2. So, this is the simple enzymatic like enzyme kinetics equation. So, if you can rearrange this, so this is like goes to this. So, I do not think you need to require to remember all these derivations, okay, but I have just given you for that if you just do it one time with your hand, okay. So, that is how you do not need to memorize the equations, you can easily derive the equation. If you just remember this particular equation, you can easily derive any formula of any Michaelis-Menten constant or so on, okay. So, that is why I am just showing you. So, to simplify the equation, so we assume that the Km, which is the Michaelis-Menten constant, as K minus 1 plus K2 divided by K1, so which yields this equation, which is our enzyme kinetic equation, okay, we all know it. So, the total concentration of enzyme, now it can be given by E0 is given by E and plus ES, which is the free enzyme concentration and enzyme substrate concentration. So, now using the equation, and if you just replace this E with this particular E0 minus ES, so we will have this kind of equation, which gives us the particular concentration of product, which is gives us this equation, okay. So, all these equations, as I already told you, you do not need to remember, but if you just do it one time, I think it will be easier to you to remember the Michaelis-Menten constant and so on, all these things, mathematical equations. So, now understanding Km and Vmax. So, Km is the substrate concentration at half Vmax, Km is the Michaelis-Menten constant, right, and it is a constant for a given enzyme. Km is the estimate of the equilibrium constant for S binding to E. The small Km means tight binding, high Km means the weak binding. Km is a measure of substrate concentration required for effective catalysis to occur, and Vmax is the constant for a given enzyme. It is a theoretical maximum rate of equation, but it is never achieved. To achieve Vmax, it will require all the enzyme molecules tightly bound to the substrate, which is theoretical, which is not possible. Okay, it is not. It is only the theoretical term. So, one more important equation which we have done many mathematics also, and also which is very important, is known as the Lineweaver-Burke plot or double reciprocal plot. The first plot which I have shown you was the Michaelis-Menten equation plot, okay, and then we have this Lineweaver-Burke plot which is called the double reciprocal plot. So, in this case what happens, like if you just check out the last equation which we have, so this is the V equal to Vmax S by Km by S. If you rearrange this equation and take the reciprocal, so you will derive this equation. This reciprocal of both sides of Michaelis-Menten equation will give you the Lineweaver-Burke equation, okay. So, in this case, if you compare it with y equal to ms plus c, so this y is 1 by v, x is 1 by s, and this Km by Vmax is the slope, and 1 by Vmax is the intersection. So, if you are given any mathematics telling you the slope and intercept of the Lineweaver-Burke plot, I think now you can easily solve what is the terms which are in the particular mathematical equations, okay. So, now, okay, before starting I just want to check if there is any doubt or query still now. So, as I have told you, if you have any problem, you can let me know in the chat box. I am keeping the chat box open always. So, if you have any problem, you can let me know in the chat box, okay. So, now drug discovery and development process. So, in medicinal chemistry, knowing the drug discovery and development process is very crucial. Like, what is the pharma drug development circle? So, first the strategic the percentage of R&D investment per load drug, this is the particular percentages of this. So, this is a strategic decision or target identification. So, at first 5,000 to 10,000 compounds are chosen at first, okay, and then from this is the period of 1 to 5 years which takes the drug discovery and preclinical phase, which sorts this particular 5,000 to 10,000 compound to approximate 250 to 5 compound. These 5 compounds, they enter to the clinical trials with the phase 1, phase 2 and phase 3 clinical trial, which takes up to 5 years to 13 years next, okay. And after this phase 3 we have the FDA approval, FDA review, and after the review we have this particular one FDA approved drug, and then we have a post surveillance market particular thing also, and then this is called the inverted pyramid of drug discovery and development. So, compound success rate of clinical trial, around 40% compounds advance from phase 1 to phase 2, one third of the compounds advance from phase 2 to phase 3, and 2/3 of compounds advance from phase 3 to FDA approval, and around 10% of the compounds entering the clinical trial stage advance to the FDA approval, okay. So, this is the particular compound success rate for clinical trials. Now, what is FDA? So, FDA we have already discussed a lot of time. It is the Food and Drug Administration and it is a federal agency of United States and of Department of Health and Human Services, which is one of the United States federal executive departments. The FDA is responsible for protecting and promoting public health through the control and supervision of food safety, tobacco product, dairy supplements, prescription and over the counter pharmaceutical drugs, medication, vaccine, biopharmaceutical, blood transfusions, medical devices, electromagnetic radiation emitting device like ERED, cosmetics, animal foods and food and veterinary products. Then we also have European Medicine Agency or EMA. Then we have MHRA, which is the MHRA is the Medicines and Healthcare Products Regulatory Agency, and it is a executive agency of Department of Health and Social Care of United Kingdom, which is responsible for ensuring medicine and medical work that are acceptably safe. Then we have PMDA, which is a Pharmaceutical and Medicinal Devices Agency, is a Japanese governmental organization similar to function of the FDA of United States, of the Medicines and Products Agency of United Kingdom, and then also the Central Drugs Standard Organization in India and European Medicines Energy in Europe. We have in India, we have two agencies, which is the one is Central Drugs Standard Control Organization under the Directorate General of Health Services, Ministry of Health and Family Welfare, Government of India, which is the National Regulatory Authority of India. And all we know about the FSSAI, I think the Food Safety and Standard Authority in India, which is for if you just buy any food component, you will see there is a FSSAI approval in that particular, which is very required. So, these are the different regulatory bodies which are responsible for various drugs and food items which we take in the market. Okay. They approve, they take care of all the safety and measures, everything they take care of it, and they approve. After their approval, any compound, any drug or any food item can be come to the market. Okay. Then I will go to so now what I will discuss is, which is in a very important topic of drug discovery and development process, which is known as assay development. So, what are assays? So, basically assays are the investigative procedures that assess a compound's effects on various biological targets. Assay development involves optimizing the condition for testing in preclinical settings. These conditions include temperature, pH, salt concentration, solubility, order of reagent addition, buffer and components required. And no drug discovery project can be successful without a proper assay. The developed assay should be relevant to the disease, and assay should give a reproducible results. So, basically assays are like that whenever you are finding out a compound, the famous assays is known as, like, there are various assays. Okay. Like I will give you just example. Let us say you have to find out what is a concentration, inhibitory concentration or IC50 value of a particular compound. For that you can just do a which is called a MTT assay. So, what happens in case of MTT assay, just MTT is a dye basically. Okay. It changes to particular color, okay, or it changes to it is generally it is pink and it forms a purple crystal which is known as formazole, and this procedure or this particular color change or this this reaction is catalyzed by the ATP in the living cell. Okay. The ATP of living cell, it is responsible for that color change. So, what happens, basically it is known as the plate reader. Okay. This is known as a plate reader. So, in this particular there is several you can see there are several wells here. Okay. And in this wells the cells are given, the particular cells are given and the compounds are given at different concentration. The compounds for which you want to check if the compound is inhibiting the particular cell growth or not. Then you just add the MTT. After incubation of few hours you have to add the MTT, and then you have to keep it for some time. If the compound which I am adding, it is killing the cells, so what will happen, the ATP concentration will be less, so the MTT to formazole formation will be less. So, that compounds will be most potent. So, that is how this is a one particular just simple example. There are lots of assay in case of the medicinal chemistry and drug discovery and development process which are required, and the assays are set in a particular for a particular disease. Okay. So, it is very highly changeable and it needs to be modification time to time. So, a bioassay is an analytical method to determine concentration or potency of a substance by its effect on living cells or tissues. Bioassays are quantitative biological assays used to estimate the potency of agents by observing their effects on a living animals in vivo or tissue or cell system in vitro. Bioassay is used to detect the biological hazards or give a quality assessment of a mixture. Bioassay is often used to monitor water quality and sewage discharge and impact on the surroundings. Okay. ELISA is very important or very well known bioassay which is known as enzyme linked immunosorbent assay. It is a quantitative analytical method that measures absorbance or color change from antigen antibody reaction, for example direct, indirect, sandwich and competitive. ELISA is used to measure the variety of substance in human body from cortisol levels for stress to glucose level for diabetes. Then we have microsomal stability assay. A liver microsomal stability metabolic stability assay was validated to evaluate liver enzyme metabolism using model compound. We all know liver is the major drug metabolisms happen in the liver. Okay. It is the most important organ for in case of the drug metabolism. In addition, the assay was evaluated by testing a set of structurally unrelated compound for metabolic stability. Other than efficacy of interaction with molecular target, metabolic stability is the primary factor responsible for the failure or success of a compound in the drug development pipeline. The ideal drug candidate should be stable enough to reach its therapeutic site of action. Despite many recent excellent achievement in the field of computational methods supporting of a drug metabolism studies, a well recognized procedure to model and predict metabolic stability quantitatively still lacking. The metabolic stability of a compound assessed in vitro incubation in presence of liver microsome and subsequently quantified by liquid chromatography, mass spectrometry or LCMS. Metabolic stability as defined by the percentage of parent compound lost over the time is assessed in the presence of liver microsome. The assays are, for example, like there is MLM, which is the mouse liver microsome, we have RLM, which have rat liver microsome, with the human liver microsome, also we have monkey liver, dog liver microsome. These are the typically microsomal stability assays which are used in the laboratory to assess the metabolic stability of a particular compound. Then one very important topic in the medicinal chemistry is the pharmacokinetics and pharmacodynamics. Okay, what is the pharmacodynamics? So pharmacodynamics is the study of how the drugs interact with a molecular target, that is the effect of the drug on the body. And the pharmacokinetics is the study that how the drug reaches to its targets in the body, how it is affected in the journey or the effect of the body on the drug. Pharmacokinetics is the study of the how the the drug is absorbed, distributed, metabolized and excreted in the body, which we all know as the famously known as the ADME. Okay, ADME means absorption, distribution, metabolism and excretion. So basically this is a very popular term which I have told you, I think, when we were discussing about pharmacokinetics and pharmacodynamics. Like pharmacokinetics is what our body is doing to a particular drug, like it is taking after taking the drug, our body is absorbing, distributing, metabolizing and excreting the drug. So pharmacokinetics is what our body is doing to the drug, and pharmacodynamics is what the drug is doing our body, how it is interacting with the receptor or the target or the enzymes and everything. Is it acting as agonist or antagonist, enhancing or inhibiting a particular biological target? That is known as the pharmacodynamic study. So these are the various phases of pharmacokinetics. The first is the drug administration, which can be oral, intravenous, intraperitoneal, subconscious, intramuscular or inhalation method. Then we have the absorption or distribution in the membranes of oral cavity, gastrointestinal tract, peritoneum, skin, skin, muscle and lungs. After the absorption, this particular drugs goes to the plasma protein binding, which goes to the neuron receptor, which can be a target site. Then it goes to the inactive storage deposit like in bone and fat, it is the binding. Okay, then it can go to the liver where the drugs go, which is the inactivation or the metabolism happens, and we get the metabolites, which some metabolites which we don't require in our body, it goes to the intestine, kidney, lungs, sweat glands, etc. for the excretion, and excretion products like urine, sweat vapor, sweat, saliva, feces. So these are the excreted products. So that's how the particular drug which is taken goes to the different phases of our body, which is known as pharmacokinetics. And pharmacodynamics, we have receptors that are the protein molecule with one or more binding site located on the cell membrane. They receive a signal from body's chemicals, neurotransmitters, hormones and enzymes. Signal will cause a molecular event on the inside of the cell to occur. The drugs enhance like agonist, diminish, partial agonist or block is antagonist to the generation, transmission or receiving the signal. Affinity, which attraction between drug and receptor. High affinity, the drug will bind easily to the receptor. Low affinity means the drug requires the higher concentration to get the therapeutic response. Drug potency is the amount of drug required to produce therapeutic response. Drug response curve, it have the effective dose ED, amount of drug that produces a therapeutic response in 50% of people taking it. We have a toxic dose, which is TD, amount of drug produces adverse effect in 50% of people taking it. Therapeutic index TI, margin of safety ratio between TD and ED. Higher the TI, the safer is the drug considered to be. In general, non-prescription drug have a higher much TIs than prescription drugs. Okay. So now these were the topics which I thought it will be better if I give you a summary, because these topics were discussed in your course, some were not very much highlighted. So I thought to highlight the some things. Okay, and then what I will now do is, so this was few topics, the rest of the topics which I think is important I will discuss on Tuesday. Okay, the day after tomorrow, the Tuesday, in our usual class, which will be our last problem solving class. The rest of the topics which I need to summarize, I will discuss on that day. Okay, so right now what I will do is, I have few questions for you for today. Like every day we do the MCQ kind of questions, so I will give you the MCQs. There will be around there will be not much, I will give you around 5 MCQs today, and like every day I will show you the answer after waiting for some time, and we will discuss that topic. Okay, but all of you are present, I want all of you to respond and give the answers, because now we are almost at the end of the session. So I think now you have to start revising the topics which you have already discussed in your course. Okay, so now let us start with the question answering session. So the first question of today's session will be, Calcium channel blockers are used for number A, hypertension and heart diseases, number B, leukemia, number C, peptic ulcer, and number D, type 2 diabetes. Calcium channel blockers are used for which disease? I will give you some time. After 2 to 3 minutes, I will check the chat box. I will show you the answer and we will discuss that thing. Calcium channel blockers are used for which disease? So, calcium channel blockers are used for which disease? Let me check the chat box. Okay, so all of you have responded as it is number A, to reduce blood pressure. Okay, let me check if it is correct or not. So, the correct answer will be, very good, it is for number A, the hypertension and heart diseases. So, what is the mechanism of action for calcium channel blockers? Okay, it has 4 effects. They act on the vascular smooth muscle, reduce the contraction of arteries and cause the vasodilation. They act on cardiac muscles, myocardium. They reduce the force of contraction of heart, the negative inotropic. They slow down the conduction of electric activity within the heart, slow down the heartbeat, and negative chronotropic. They block the calcium signal adrenal cortex cell. They directly reduce the aldosterone production, which correlates to lower blood pressure. The therapeutic uses of calcium channel blockers are the angina pectoris, which is the verapamil diltiazem, cardiac failure, the verapamil diltiazem, amlodipine, nisoldipine. Then these are the hypertensions like verapamil, migraine for verapamil, stroke for nimodine. The side effects are the tachycardia and orthostatic hypotension. So, these are the side effects of the calcium channel blockers. Okay. So, just remember what is the mechanism of action of calcium channel blockers and also the therapeutic uses, okay, where it uses and what are the names of few calcium channel blockers. Like the question can be given like verapamil is which kind of drug. Okay. So, what is the mechanism of action? So, then you have to answer it like it is the calcium channel blocker, right. Okay. So, now I will go to the next problem. So, it says that nevirapine is a drug which is a dash inhibitor of reverse transcriptase. Your options are number A, allosteric, number B, non-nucleoside, number C, non-competitive, number D, all of these. Nevirapine is a drug which is a dash inhibitor of reverse transcriptase. Your options are allosteric, non-nucleoside, non-competitive, and number D is all of these. So, which of the following will be the correct one? Think and then let me know in the chat box. So, nevirapine is which kind of inhibitor of reverse transcriptase? Let me check the chat box. Number C, number B, number B. So, I have one number C and number 3 number B. So, only 4 answers. So, why others have not responded? I want all of you to try, okay? Even if your answer is wrong, it does not matter. But trying is very important since we are almost at the end of our this course. So, now I think all of you try to answer the questions. I will give you 2 more minutes, then I will show you the answer. Okay, let me check it once more. Okay, number B. Okay, let me check if number B is the correct one or not, or number C is the correct one. So, the correct answer will be number D. All of these are correct. Okay, this is the structure of the nevirapine. So, the nevirapine, it is known as the non-nucleoside reverse transcript inhibitor or NNRTI. So, the mechanism of action is like they act by stopping the HIV production by binding directly into the reverse transcriptase non-competitively and preventing the conversion of RNA to DNA. They do not require activation through the phosphorylation. The non-competitive inhibitor, the non-competitive inhibitor is distinguished from general or mixed inhibition from the inhibitor that has an equal affinity to the complex. Okay, the enzyme and substrate, there is an inhibitor. So, in non-competitive inhibition, inhibitor binds to the site other than the active site, that is known as the allosteric site. Okay, so in case of non-competitive, wherever there is a non-competitive inhibition, there the particular compound does not bind to the active site where the substrate bind, but otherwise it binds to the site where the other side, which is known as the allosteric site. For example, this is an enzyme, this is the active site where the substrate binds generally. Okay, and this is called the allosteric site. So, if the allosteric inhibitor or non-competitive inhibitor binds to the allosteric site, so what happens? The active site shape changes. Okay, now the substrate cannot bind to the particular active site of enzyme, and that's how the inhibition occurs. Okay, for example, there is a pyruvate kinase is here. Okay, and there is a your alanine. Okay, and there is a phosphorylation happens in case of the reaction. Okay, and then if the changes of amino acid happen, so this catalysis cannot happen. The amino acid in non-competitively binds to the enzyme kinase here. Okay, so this is this. So remember, nevirapine is allosteric, non-nucleoside, and also non-competitive inhibitor of reverse transcriptase. Okay, the three, all of these are correct. So now next problem, it says which of the following is the most important enzyme is in the cytochrome P450 family. Your options are number A CYP1A2, number B CYP2C9, number C CYP3A4, and number D CYP2D6. Which of the following is the most important enzyme in CYP450 family? Which of the following is the most important enzyme in CYP4550 family? Let me check the chat box. Number C, number C, number D, number C, number C. So I have number C mostly, but one number D also. Okay, let me check which of the following is the correct one, number C or number D. See, even if your answer is wrong, it doesn't matter, but answering is very important, which you are doing, which is very good. So the correct answer will be number C, CYP3A4. It is the most important enzyme of CYP450 family. So the drug metabolizing enzyme, so liver is the major organ for the drug metabolism and elimination. The phase 1 and phase 2 enzymes, phase 1 enzyme oxidative and hydrolytic reactions happen. In case of phase 2, the conjugative reactions happen. Predominant enzyme system that metabolizes the drug is the cytochrome P450 family of the enzymes, which mediate oxidation reactions such as hydroxylations. So there are different enzymes which are the important in case of the CYP450 family. There we have the CYP1A2, which known the drugs to be metabolized is the 4%. So 4% of known drugs are metabolized by this enzyme. CYP2C9, 10% of drugs are metabolized by this enzyme. 2% of drugs are metabolized by CYP2C19, 30% of the drugs are metabolized by CYP2D6, and 50% of the drugs, most of the known almost 50% of known drugs are metabolized by CYP3A4 enzyme. The role of the CYP enzyme in hepatic drug metabolism, the relative hepatic content of CYP enzymes is the there is different concentrations, okay, and this is the pie chart for the percentage of drug that is metabolized by CYP enzyme. So remember, CYP3A4 is the most important enzyme of cytochrome P450 family. So now next problem, it says that statins work as number A, HMG-CoA synthesis inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthesis inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. Statin works as which kind of inhibitor? HMG-CoA synthetase, HMG-CoA reductase, squalene synthesis, or acetoacetyl-CoA thiolase inhibitor. Let me check the chat box now. number B, number B, number B, number D. HMG CoA is the largest number, second largest is the metabolism, second largest is the, yeah, it is the correct, second largest is the CYP2D6. HMG CoA reductase inhibitor, D, number B. Okay, so I have number B and number D as answers again here. So let me check. So correct answer is yes, number B, HMG CoA reductase inhibitor. So HMG CoA reductase inhibitors are the particular, which as the example is the stannin. So mechanism of action is they are the analog of HMG CoA. These are the competitive inhibitor of HMG CoA reductase. The rate limiting step in cholesterol synthesis, reduced hepatic cholesterol results in the increased LDL or receptor expression, which increases the removal of LDL from blood, and reduces hepatic cholesterol, result in decreasing the VLDL in the synthesis and secretion. The adverse effects are hepatotoxicity less than 1% of patients, myopathy 2 to 10% of patients, rarely leads to rhabdomyolysis and contradicted in pregnancy. So these are the adverse effects and these are the mechanism of action of HMG CoA reductase, which are the stannin. So these are the very important medications in case of the cholesterol management. Okay, statins are the very important drugs in the cholesterol management. The famous cholesterol managing drugs, you will if you go to the market, if you have anybody of your known person, if they have cholesterol, so if you stay there, look at their medicines, you will say there is the there is a suffix of statin, like atorvastatin and this like of drugs. So the drugs, the names are at last there will be a statin. So you will understand that are the HMG CoA reductase inhibitors. Okay. So now the last question of today's session I will show you. It says that the enzyme which catalyzes the fibrinogen to fibrin conversion is number A, trypsin, number B, thrombin, number C, prothrombin, and number D, pepsin. So which of the following enzyme catalyzes the fibrinogen to fibrin conversion? Let me check the chat box now. Yeah, pepsin convert the protein in smaller proteases, very good. Trypsin convert protein into amino acid, that is also correct. So what is the correct answer? It is the thrombin, number B, number B, number B. Okay, number B. So most of the answers are given as number B. Let me check if number B is the correct one or not. So the correct answer will be yes, very good, number B, thrombin. So thrombin catalyzes the fibrinogen to fibrin. So basically it takes part in case of the your blood clotting, right? If you have any cut or anything, so that there is a the blood, after some time the blood gets clot, okay. So that is the whole mechanism. So first is there is a prothrombin which converts to thrombin, and this is catalyzed by a prothrombin activator and calcium ions, okay. Calcium ions are very important in case of blood clotting. This thrombin catalyzes the conversion of fibrinogen to fibrinogen monomer to fibrin. Here also the particular calcium 2 plus ions are involved, and thrombin also give rise to the activated fibrin stabilizing factors which converts the fibrin from fibrin fibers to the cross-linked fibrin fiber. So basically if you have any cut or any particular injury, so what happen, there is a platelet aggregation happens, okay. In case of this platelet aggregation, so what they will do is they will activate the coagulation. This coagulation or this particular coagulation goes to the give rise to the thrombin. This activation goes down to the thrombin which convert the fibrinogen to fibrin. The first is the clot initiation, then we have this thrombin which give rise to the fibrin and fibrin to retraction, and the blood clot is formed here, and thrombin fragments are formed by the fibrinolysis, okay. Now after the skin is these things is healed, then the fibrinogen fragments happen, which is known as the fibrinolysis. And sometimes you will see that there is a particular the blood clotting happens even without injury. In that case blood thinners are given, so that this particular so that the clots do not happen without any injury, or in case of in the normal blood flow, okay. So this was the last question of today's session. So today what we had do, that we had discussed the topics of medicinal chemistry which were important in your topics, I thought, which were not gravely discussed in your course, and then we will solve few questions. So I have checked that if you are reading things or not, because now we are almost at the end of our session. So like always I will share my email ID here at the chat box, okay. If you have any query or any doubt, okay, first let me check share my email ID. So if you have any problem, any doubt regarding any topic of medicinal chemistry, please let me know by tomorrow, okay, because day after tomorrow on Tuesday we will having our last problem solving session. So if you have any problem or any doubt, you want me to repeat any topic, I will try to repeat that on that day, okay. So all this lecture video and presentation I will share to you. I know, and now I think you know how to I mean download this videos and the presentation, right. Last day after the class I have shown you. What you have to do is, I will just show you one more time where to find this problem solving session video. So what you have to do is, you have to go to the SWAYAM portal, okay. Just let me show you how to do it. So first you have to go to the SWAYAM portal. Second, yeah. So you have to go to the SWAYAM portal at first in the medicinal chemistry course, okay. I think all you are having this kind of emails, which who have registered for the course, you are having this kind of emails, okay, like for the lecture videos and also for the all these assignments and things. So if you go to the SWAYAM portal, first you have to log in using your credentials like your email ID or everything. I will show you where to find this particular lecture solving videos and also thing. Exam pattern, which topic should we focus more? Okay, the exam pattern, it should be both subjective or objective based, okay. The topics which I think most of the you will find whenever you are going through a course, the topics from which the questions are given in case of your assignment, okay. The topics for which are more given questions from the assignment, I think you should focus on those topics more, okay. Okay, after opening the medicinal chemistry, you will have at the left side, this is your week wise assignment and your top videos linked. At the end you will find a problem solving sessions, and if you go to that, there is a there is a one is problem solving session recording. If you click on that particular problem solving session recording, it will give you a particular Excel sheet. In this Excel sheet you will find the YouTube links of the all lecture videos of the past classes and also the PPT link. If you click on the link and the video, you will find it there, and also there is a particular problem solving session survey which is a Google form. So I wish all of you whoever have attended the medicinal chemistry course, just before we are ending the course, please open this Google form and just give a feedback on this problem solving sessions. Okay, that will also help me to understand what things you need, so I will just try to improve myself from the next semester. Okay. So now today it was, so I think the topics which are given in the assignments, you should focus on the topics more. Okay, I think it will be sufficient. And also if you have any problem at any topic understanding, please let me know in this email ID by tomorrow, so that at the last class, which will be on day after tomorrow, Tuesday, I can discuss that topic. Okay, if you have any doubt, any query, just let me know in that email ID. So is there any problem or any topic you want to tell me right now? I have a couple of minutes left. The last class, I want all of you to attend the last class, so positively, which will be on Tuesday. Okay, we will have our usual 2-hour class on that day. First hour I will discuss the topics, I will summarize the topics which are important. I will basically on that day I will discuss about various drugs like antibacterial, antiviral and anti-cancer and so on. And after that I will show you around 12 questions from 12 weeks. Okay, each week one question like this, and I think all of you can answer those questions now correctly, because we are at the end of our medicinal chemistry course. So if there is no problem left, I will close the session for today, or if there is any problem, you can let me know right now. If not, then I am closing the session for today. Just note the email ID. Okay, and if you have any topic you want me to repeat, please let me know by tomorrow. Otherwise it won't be possible for me to prepare the slides for your class. Okay, so I am closing the session for today. Thank you all for joining. I will share this lecture presentation, and also I will share the video of the today's class soon in that particular Excel sheet. Yeah, thank you all for joining, and all the best for the examination. Let's meet on our last problem solving session on day after tomorrow, Tuesday, from 6 p.m. Okay, all of you please join on that problem solving session positively. So I will close the session for today. Thank you all.